cockroaches, dragonflies, and damselflies. And she says that entomologists are nowhere near completing an exhaustive study of all the insects on Earth. And now we may not have a chance to do so before species disappear.
You are probably familiar with the plight of the flight of the bumblebee. Bumblebee populations have been in decline, and for the first time, a bumblebee has been put on the endangered species list.
But the recent study carried out in nature preserves over 27 years by German researchers found a major decline in all flying insects. Now, the insects were measured by weight, and the collective biomass fell by 77%, and in the summer, when insects are the most numerous, by 82%.
Dr. Ware reminds us that the members of the following list, which are just a tiny fraction of all insects, are animals we cannot live without. So let us give you, for starters, the beetle, the bee, the butterfly, ladybugs, bedbugs, praying mantis, aphids, earwigs, crickets, dragonflies, stink bugs, and kissing bugs. What do they all have in common?
They're all members of the class Insecta, the most diverse group of organisms on the planet.
I see. Okay, now, you were part of a team of about 100 scientists that helped to map the evolution of insects a few years ago, and the result being the first phylogenetic tree of insects. That is a tree showing evolutionary relationships. Okay, well, what was the first insect? How far back do these guys go?
Well, we know that winged insects are about 406 million years old, and they probably looked something like a dragonfly. Maybe they were larger than the dragonflies we see today, but they probably looked something like that.
Okay, so what niche did insects fill? I mean, that's a long time ago, 400 million years ago. I mean, that predates the dinos and so forth. They must have seen an opportunity. There must be some, I don't know, evolutionary niche that wasn't so niche-y for this kind of an animal.
Well, certainly the first things to take to the skies were insects. So before insects started flying around up there was nothing there. There were no birds, there were no bats, there were no pterosaurs. So that's a wide open niche space to exploit. And then most of the terrestrial habitat had not yet been exploited by other animals yet. So insects were able to fill basically all of the terrestrial niches and the air.
They're also small, at least they seem to be small today. Have they always been quite so small?
No. In fact, some of the ancient relatives to modern insects and modern arachnids were much larger. There were some scorpions that were the size of a terrier. There were these things that were close relatives to modern dragonflies, but not actually modern dragonflies. And they had wingspans of, each wing was about 37 centimeters, which is about 3 feet long. So those are big to have those flying around above you.
My goodness, that sounds scary. So I've heard it said that insects can only get so big because they don't breathe through a nose or their mouth or anything like that. They breathe through these little holes in their hard outer skeletons, and consequently, if you make them too big, then it's too far from the air to go into these bodies. Is that the deal? What limits their size?
Well, certainly the spiracles, which are the organs you're talking about, these little holes in the side of their body, which through air enters the body, and their circulatory system is pretty rudimentary, so it's very hard to have them be extremely large and to be able to be fast, to be able to do things that require a lot of oxygen transport and a lot of blood transport. But in addition, the larger you are, the easier it is for you to dry out if you have a large surface area. And insects that have just soft bodies, a lot of them have just soft bodies, they would dry out very quickly. So a lot of the times when we imagine what these giant things must have looked like during the Carboniferous period, they weren't necessarily the fast-flying, quick-turning dragonflies that we see today. They would have been kind of slow-moving, kind of with wings that paddled through thick, viscous air, almost like paddling a canoe through water. And they would have been probably pretty clumsy. But certainly as birds came on the scene, we see dragonflies modified the way that their wings are shaped and the way that they fly. So they suddenly got very good at getting away very quickly. And presumably that had to do with the pressure of predation, but also with the fact that they had birds competing with them for the other insects that they were eating.
When did insects first start to fly? Did the first insects fly, or were they just, relatively lightweight and they could, hop big distances and eventually started, flailing their legs? Or how did that happen?
Oh, that's a hot debate. In the entomology world, how insects first started to fly is a really hot debate. And different things have been proposed, from starting to glide slowly, from hopping around, from insects that have aquatic life stages, that have gills on their abdomens that may have been modified to be wing-like. People have debated about this for decades. But what we know is that there are insects that we call the basal hexapods, and they are insects that do not have wings. They first speciated before the first flying insects. And they all are very small-bodied. Most of them live under the soil, and they don't have a lot of, they're not very charismatic, put it that way. And they're not very colorful, kind of drab-looking. There's not very many species present in those taxa. Once insects evolved wings, once the winged kind of body type evolved, from that point onwards, that's when we really see insects rise to being the dominant, species that we know of when we think about biodiversity.
Well, Lauren Esposito from the California Academy of Science is here in the studio with us and with a bunch of cockroaches in a what I hope is a well-sealed container. As we look at these insects up close, at these little roaches scurrying around here, is there anything that we should pay attention to that will perhaps help us appreciate it somewhat better?
Well, when we think about cockroaches, we always think of them as being really vile and dirty. But what you might notice when you look at the cockroaches is that they spend a lot of time cleaning. So you'll see that they'll take their antennae and they'll drag it, over their mouth parts, holding it with their feet, their tarsi. And what they're doing is actually keep their antennae very clean so that way they can use their antennae to kind of sense around. There's 4,500 species of true cockroaches plus another 2,900 that are termites. 2% of those are pests. The rest of them live fantastic lives out in the jungle or in the forest, really not carrying a lot of disease as far as we know. It's just those 2% that are living in the sewer that kind of give the rest of the cockroaches the bad name of them being kind of dirty.
Jessica, something is happening to flying insects. I believe the alarm started in earnest with a German study released in 2017 that found that over the course of nearly 30 years, there'd been a major decline in flying insects in one region. Now, can you tell us about this study? What's going on here?
This study was a really interesting study that harnessed something that we all have access to, citizen science data. So they used citizen science data that had collected insects, doing bioblitzes and different things like that, and they weighed the mass of insects that were collected each year in these different sites in Germany. And what they found was that the overall mass of insects that they were collecting was decreasing over time. And what that could suggest is that insects are getting smaller, but it could also suggest that there's fewer insects in general that are being collected despite the same amount of human effort. That's alarming because what we've noticed is beyond that study, in monarchs, number of monarchs that are showing up at their migration habitats are also decreasing. Number of mosquitoes are decreasing. So it's a trend that we're seeing in many different studies for many different groups of insects. The number of individuals is going down.
What's the theory about what's going on here? I mean, bumblebees began to decline in 2017, apparently. Bumblebees were placed on the endangered species list for the first time. I take it that's not related to the other kind of bees that have been declining, the collapse of the honeybees.
Well, in general, a lot of the factors that are causing, these different insect groups to decline are similar. So loss of habitat is something that is common for all of these insects that we're talking about. The use of neonicotinoid insecticides has been implicated for bees, but those neonicotinoid insecticides, which linger in the environment, can also affect different types of insects, right? From butterflies, like monarchs, to other types of insects. And in addition, the climate has been changing. So we've seen erratic, variable seasons where seasons end up being drier or wetter than what is normally expected for that time of year. And all of these factors together creating these urban deserts by having, a reduction in habitat, having the habitat be poorer in terms of the quality of plants that are there, the presence of insecticides, and the unseasonable weather. All of these things combined can dramatically affect whether or not insects survive to reproduce and lay their eggs so that they can have progeny the next year.
Well, finally, Jessica, I know this is probably an impossible question to answer. Nonetheless, I will ask it of you, and that is, can you envision what life would be like in a world without insects? I mean, if all the insects were to go away, what would life be like on Earth?
Well, for humans, it would be pretty much uninhabitable. When you think about it, in our day-to-day life, we obviously know that we rely on insects for pollination and for food. So we would imagine a future without fruit, without coffee, without blueberries. Those things I think have already been well covered by the media. But there's other things that we rely on insects for that we kind of take for granted. So heaven forbid we talk about death, but death is something that comes for all organisms on Earth. And there's insects that do a service, that are decomposers, that basically decompose all of the dead things, whether it be dead trees, dead plants, and dead bodies. Without insects, those bodies would not decompose. They would decompose at a very slow rate, perhaps just by fungus and microbial decomposition. That would be very, very slow. So we would have kind of littered carcasses of plants and trees and animals all around without insects to do that work. As well, without insects, they're the main food source for a lot of birds, for a lot of bats. So if you imagine a world without insects, you imagine a world without birds. You imagine a world without bats. You imagine a world without lots of organisms that rely on insects as food. So the type of habitat that you would picture would be one that would be very different in terms of the types of plants that would be able to survive and the types of animals that would be able to survive. We certainly would not.
Jessica Ware, thanks so very much for speaking with us.
Thank you for having me.
Jessica Ware is an evolutionary biologist and entomologist at Rutgers University, Newark.
Lauren, where did these cockroaches come from that we have here in the studio?
Well, these particular ones came from a colleague's apartment where they normally live in the bedroom of her children.
What?
Yeah.
But in a container, or they were running around the house?
Usually they're in a container. Because these species were originally from Madagascar, they're really incapable of surviving outside of a human house. They need it warm, they need a food supply. So these ones normally live in a cage, and they sleep on the shelf of a 11-year-old girl.
Can we touch the cockroaches?
Yeah, sure. We can pick one up and take it out, and we might get lucky and be able to hear it hiss.
These guys, they don't know what's about to happen to them. But then again, either do I.
Okay, so Lauren is opening the cage.
Opening the lid.
Oh, goodness.
Blow the lid off this show, okay? All right, I got my hand out. It's just like Halloween, except they're going to put a bug in it.
Did you hear that hiss?
Wait a minute.
It sounds like air leaving a balloon.
That's kind of the idea. They're pushing this air out of their spiracles, which are their lung openings, and it's meant to ward off predators.
Little cockroach buddy.
Boy, it really is hissing. Is it hissing because it's mad?
I would say scared.
Okay. Oh, he's incredible. The legs feel like they're strong. It's kind of tickly. He's actually quite gentle. He looks like he's a little afraid, which he is.
Is this the heat?
That is the heat, yeah.
I never thought I would hold a cockroach willingly. And look at him. This shell is really extraordinary on the back of this cockroach. What can you say about it?
So these cockroaches, they're really like a rich chestnut fading into black color, and it's really shiny, thick shell. Shell's an accurate word to describe them. They're related to crustaceans like shrimp and lobsters that we consider to have a shell. And he's perfect for blending in, and he's got this armor, this leathery armor, to keep things from eating him, to keep him protected. And
what do you think he's gathering about being on the palm of my hand? First of all, it's quite bright, but is he picking up chemical signals from my skin?
Sure, he's definitely smelling the environment using his antennae, and he's also feeling with those same antennae. So he's just trying to figure out where he is. It's kind of an unusual circumstance to be held by another thing.
Let me give him back to you. Lauren, are you willing to hang on a little bit longer?
Absolutely.
Okay. And are the cockroaches happy?
As happy as they can be, I suppose.
Can you tell when they're not?
Well, they start hissing.
Coming up, high-tech tools may provide an efficient way of getting rid of malaria. But are we ready to actually tweak the genome of the mosquito to do it? Using CRISPR and gene drives to rid ourselves of a deadly pest.
It's the X-Flies on Big Picture Science.
We're appreciating the diversity and evolutionary adaptations of insects, including the hissing cockroaches that are right next to me here and who are auditioning for a part in the show. Insects are indispensable to our ecosystem, and their disappearance is alarming. However, there is one species that might be universally voted off the island.
Mosquitoes do have ecological niches as pollinators and as a source of food for birds and reptiles. But more than any other insect, they are notorious vectors of disease. The mosquito is small enough to perch on your fingernail, yet no insect species has been a more efficient killer of humans.
The mosquito species Aedes aegypti can carry yellow fever virus, dengue, and Zika. Many mosquito-borne diseases are caused by viruses. Malaria, however, is caused by a one-celled parasite and is carried by 40 species of the mosquito genus Anopheles. A person who falls ill with malaria may feel like they have the flu, but left untreated, the illness can lead to complications and death. According to the Centers for Disease Control, in 2016, there were 216 million cases of malaria worldwide, and almost 500,000 people died, mostly children.
Scientists are applying high-tech tools to fighting the disease. This includes the new gene editing technique CRISPR, which allows for precise and rapid edits, combined with gene drives, which greatly increase the chance that genetic modifications are inherited over and over.
Hi, this is Anthony James. I'm a vector biologist working at the University of California, Irvine, and I work with mosquitoes and mosquito-borne diseases.
His team's focus is the Anopheles gambiae mosquito, which carry not all but a large number of the deadly diseases. However, with CRISPR and gene drive technology, unforeseen consequences are a concern, and so are those anticipated. Could engineered mosquitoes create parasites or gene drives that are resistant to the modification? Dr. James describes where the deadly parasite calls home.
It turns out that the parasite can only be one of two places. It can only be in the mosquito or in the human being. The parasites feed on human red blood cells, and when the mosquito takes a meal, looking for that protein-rich meal to make her eggs, she takes up the blood with the parasites and picks them up at that point.
All right, Tony, so CRISPR, that's a genetic engineering tool, allows you to make quick and precise edits in a genome. Which part of the genome are you targeting in your battle against these malaria-causing mosquitoes?
So we're not actually editing the mosquito genome per se. What we're doing is we're using the technology to bring in a brand new gene, and this new gene has the ability that when it's turned on and working, of actually being able essentially to kill the parasites. So
you're making mosquitoes, in other words, that have the ability to kill the parasite if the mosquito bites somebody who's infected. How does it do that, actually? It turns out that there are a lot of different of these malaria parasites, and for example, the ones that infect humans will not infect mice. So people working on vaccines were able to put human parasites into mice, and mice would fight them off. And we took a part of the mouse immune system that is able to kill the human parasites and put those into mosquitoes. These modified mosquitoes would have part of a mouse's immune system. Correct. Just a tiny little part, one or two genes at most. I see. So to be clear then, the genetic modification is not to eliminate the mosquito. The mosquito probably doesn't know what's happened to it, probably doesn't feel that anything's happened to it, but it's targeting the plasmodium that causes malaria. That is correct. And so our idea is that it's going to be probably impossible to get rid of all the mosquitoes. And so we say, well, it's the parasite that's a target. So we use the mosquitoes to go after the parasite. The World Health Organization says that increased prevention and control measures already in place have led to a nearly 30% reduction in malaria mortality rates since 2010. In other words, we do seem to be moving in the right direction, and this might be used as an argument that current control measures, which include, maybe vaccines or, in any case, better mosquito nets, are working. Why do we need a genetic engineering approach? So that's a great point. And it turns out that if you look at the data, that is correct. Over the past 10 to 15 years, there's been a really remarkable and laudable reduction in the amount of malaria. However, we've seen over the last couple of years that we've bottomed out, so to speak. It's not getting any better. So we still need techniques to push this all the way to zero. Gene drives. Gene drives that's a scheme made possible, I take it, by the CRISPR technique. How does a gene drive work, and how do they ensure that, anything that you modify, its descendants are still subject to that modification?
Right. So this is an interesting question to try to answer over the radio, because I usually wave my hands around. But the idea is that there's a technology, which is the CRISPR technology, that allows you to break chromosomes. And when the chromosomes are broken, they try to repair themselves. And so what we do is we use the CRISPR technology to break the chromosomes at a very specific place, and then we add a whole bunch of DNA and hope that in the process of the DNA fixing itself, it copies some of the DNA that we put in. So we can actually make that happen. And when we do that, this process becomes repeatable. And so what we put in has the ability also to continue to cut the chromosomes and add things in.
This isn't like changing a trait for the next generation, is it? I mean, if you could modify my genome so any kids I'd have would be blondes, that modification would be washed out a few generations down the line. That's obviously not happening in a gene drive.
No. So the idea is to have it persist in the mosquitoes for, say, 5 to 8 years, long enough to have an impact on malaria. But we're not looking for an evolutionary change, something that's going to be around for 10,000 years or something like that.
When you use this gene drive, okay, you modify the DNA of a whole bunch of mosquitoes in the labs, but this isn't like the technique where you, sterilize all the males and hope that they mate with a lot of the females. I mean, that gets washed out. This obviously doesn't get washed out because it gets passed down from generation to generation. How does that work?
The idea is to have the gene drive system put into what's called the germline. So these are the sperm and the eggs, and those are the cells that contribute to the next generation. So we see that everywhere. It's the same thing in humans. They have sperm and eggs, and a sperm fertilizes an egg and gives rise to a new individual. So if our genes are in the sperm or the eggs, then the offspring that come from that will have the gene, and it'll just continue that way. So it'll be something that is heritable over hopefully at least 5 to 8 years.
So, in some sense, you've actually made a new species of mosquito.
Well, you think about it like adding 2 or 3 additional genes. And so it's not a different species, because there are natural variants of malaria vectors, of malaria mosquitoes, that are resistant. They have their own genes that help them fight them off. But it's hard for us to work with those. And so we just made some essentially synthetic genes and put those in.
Okay, designer mosquitoes.
You've got it.
Okay, Tony, well, give me the big picture of how this would work, assuming all goes well. You're going to engineer some mosquitoes in the lab, breed some nominal number of them. I don't know what that number might be. I mean, 10, 1,000, 1 million, and then release them into the wild, right?
That's correct. The idea would be that they would be part of an integrated program. Hopefully we have people using bed nets and availability of prophylactic drugs and maybe down the line a vaccine. But what we offer is a circumstance when we put the mosquitoes out there and malaria is eliminated from that area, it can't move back in. And it can't move back in because if somebody brings in the parasites, the local mosquitoes are unable to transmit. So what it allows us to do is consolidate our efforts. We can clear an area and have it remain free of malaria and move on to the next place. And so our big contribution is the sustainable aspect, sustainable consolidation of elimination of malaria.
I see. How many mosquitoes do you need to breed in order to do this effectively out in the wild, not in the lab?
Amazingly, it's not as many as you think. We imagine that individual releases will be somewhere in the range of 100,000, and we easily have 100,000 mosquitoes and many more actually growing in our insectary right now here at UC Irvine. So the idea would be probably you'd be using millions over a year, but it's easy to make that many.
Mosquitoes are easy to make, I take
Well, you just give them something to eat, and then the next thing you know, you got a lot of them.
Well, okay, about some concerns here. This technique might work in the lab, but there are questions about its effectiveness in the wild. One of the big challenges seems to be the potential that the insects will develop a resistance to gene drives, or that maybe the parasite would develop a resistance to the altered mosquito.
Correct. And so for the parasite, it's very much like a multiple drug use. So we have agents now, for example, that will cause infections in people and are resistant to antibiotics. And so what the physician will do will prescribe a combination of antibiotics. So the idea is that if one doesn't get him, the other will. And so we do the same thing, putting these genes in the mosquito. We have at least 2 genes that go after the parasite. But then for the mosquito becoming resistant to it, the good news about the CRISPR technology is that if a particular region in the chromosome is resistant to our genes, we just go to another place. And so we can overlap places where we go, so we get complete coverage, even though in one individual mosquito, it may have one resistance gene.
This sounds like the kind of cocktails that are made certain diseases.
Yes, that is exactly right. And that's the idea, is that you have multiple systems, built-in backups, so to speak. Another
concern, Tony, is that of unforeseen consequences. I think every time you talk about modifying biology somewhere, a lot of people get up in arms, remember the rabbits in Australia or something.
Right.
I mean, how can we be certain that changing this gene that stops the transmission of the malaria parasite won't also alter, the mosquito's behavior? What if the gene has other adaptive functions, or even worse, the gene triggers a cascade of troubling ecological consequences?
So we can never really know without testing whether or not these things are going to happen. But we can make really educated guesses. And indeed, because it's a genetic engineering, we can structure these DNA molecules that we put in a way that they have the minimum amount of what we would call off-target impact. And so part of our strategy of developing something for the field is to minimize the potential for these off-target effects.
Well, finally, Tony, if we succeed in wiping out the malaria parasite, will we then be able to find it in our hearts to like mosquitoes?
I think we're going to have issues still, because while we may have solved the problem for humans, mosquitoes also transmit a number of diseases to animals, and, for example, are responsible for the devastation of the native birds in the Hawaiian Islands. And so I'm hoping that our technologies will also be applied in a situation where we can do something for island conservation.
Anthony James, thanks so very much for speaking with us.
You're welcome. Thanks for having me.
Anthony James is a vector biologist at the University of California, Irvine. Well, Anthony James talks about the use of genetic technology to control malaria. Lauren, as a scientist, what is your response to the potential for that technology to eradicate a disease or even to change a species?
The idea of figuring out ways to control the diseases by controlling the things spreading the disease is not a bad idea. It would be huge for human health and human medicine. The things that I worry about are whether in changing the genetic code of these species of mosquito, so that they're no longer transmitting disease or so that their populations are limited, what happens if that change somehow makes it into other native mosquitoes that are natural to that environment or to that geographic place, and thereby reduces the population of those mosquitoes? And the reason that I'm worried about that is because many things on Earth are highly dependent on mosquitoes and mosquito populations.
Biology is so complex. Everything is so interdependent.
I mean, I think pertaining to insects specifically, the point in time when both flowering plants and flying insects evolved, there became this huge explosion of interdependence, perhaps in ways that became more and more complicated over time than they had ever been in the history of Earth. And that's where we stand today. And we're really at a point in time where we have this remarkable interdependence of life on Earth that's complicated in ways we can't even imagine.
Well, what we've heard in the show is that insects are not only interesting, I don't imagine that surprises anybody, but that they're essential and they depend on us. Yes, some of them, like mosquitoes, but we depend on them, and in ways that I had never suspected. And even cockroaches are kind of cute and appealing when you get up close and personal with one.
Lauren, thank you so much for coming to the studio and bringing the Madagascar hissing cockroach family.
It was my pleasure.
Lauren Esposito is an arachnologist. That means she studies spiders and scorpions and things like that. She's from the California Academy of Sciences in San Francisco. Well, thanks to the team members who are faster than a cockroach scurrying under your refrigerator, Senior Producer Gary Niederhoff, Operations Manager Barbara Vance, and Intern Anna Katrina Hunter.
Thanks also to financial support from Rena Shulsky-David and Sammy David, and to the William K. Bowes Jr. Foundation. Big Picture Science is produced at the SETI Institute, a nonprofit scientific and education organization whose scientists study the origin and nature of life, including the chemical history of Martian soil. And a big thanks also to our listeners.
Your ears have been attuned to an episode of Big Picture Science entitled The X-Flies. If you'd like to hear more Big Picture Science, we'll just buzz on over to our archive at bigpicturescience.org.
Did you wing that?
Find the Seven Sisters, also called the Pleiades, that most striking, dense collection of stars in the eastern sky these nights? There's nothing like it, and it's stunning through ordinary binoculars. Right now, let's talk about an invisible object near the Pleiades, the Crab Nebula. This story really started in 1054, when a new dazzling star abruptly appeared in the sky. Its glow was not starlight, nor reflected light. Its origin is neither heat nor nuclear energy. Even today, after the biggest fireworks are long over, the remaining glow of the Crab Nebula is an exotic phenomenon called synchrotron radiation, produced when electrons are forced to change direction by super powerful magnetic fields. But this strange lavender glow is just the picture frame that surrounds the inner sanctum, the object that sits at the heart of the nebula that creates the magnetic field is the Crab pulsar. A pulsar is a tiny, solid star, a neutron star, which is the original giant star's core. When the rest of the star went kablooey and exploded outward, this tiny remnant went the other way and collapsed inward. It dramatically shrunk until the material of 500,000 planet Earths packed itself into a ball smaller than Brooklyn, while its rotation sped up. These days it rotates 30 times a second. Dense or hard are understatements. The density of crab material would be exactly duplicated if a cruise ship could be crushed down to the size of the ball in a ballpoint pen. Except here, there isn't a tiny speck of the stuff, but a sphere of such impossibly packed material 12 miles across. This is the same density of every atomic nucleus in your body, though of course your body has only teensy specks of the stuff. Most star surfaces are gassy, but unimaginable gravity has forced the Crab pulsar's broken atom fragments into a solid with 100,000 trillion times the stiffness of steel. It doesn't need to be insured. You couldn't scratch it with a hydrogen bomb. For Strange Universe, I'm Bob Berman.
Dr. Seuss stops a gas pipeline. That story and more on H2O Radio's weekly news report. I'm Jamie Sudler.
I'm Franny Halperin, and it's This Week in Water.
The Paris Agreement on Climate Change was kept alive at the UN conference called COP24, which concluded Saturday in Poland.
Delegates from 200 countries produced a 156-page rulebook about how nations will report their progress to limit carbon emissions.
There was concern that the U.S. would weaken the agreement. However, the New York Times reports U.S. negotiators worked constructively behind the scenes with other countries, including China, on transparency rules.
Highly visible were the many young people in attendance. 15-year-old Greta Thunberg from Sweden admonished delegates, saying the planet was being exploited by the wealthy few.
Our biosphere is being sacrificed so that rich people in countries like mine can live in luxury. It is the sufferings of the many which pay for the luxuries of the few.
Investors agree that urgent action is needed. According to The Guardian, some of the world's biggest pension funds, insurers, and asset managers issued a stark warning that without cuts in carbon emissions and the phase out of coal, the world faces a financial crash several times worse than that in 2008.
The Congress passed a massive farm bill last week with many environmental provisions, including increased funding for a program that prioritizes projects to protect drinking water and reduce harmful toxic algae blooms.
As Mother Jones reports, the bill also incentivizes farmers to use environmentally friendly soil management practices like reduced tillage, planting cover crops, and crop rotations in a way that sequester carbon.
Farmers will be able to get insurance when planting cover crops crops, which had in the past been problematic.
Another part of the bill allows farmers to grow hemp, taking it off the controlled substances list where it had been alongside drugs like heroin.
Early drafts of the bill included a rider that would have stopped local governments from regulating or restricting the use of pesticides in places like parks, playgrounds, and schools, but that provision was removed from the final legislation.
The organization Food and Water Watch applauded the bill, which makes important progress in organic food programs, including funding for research.
The bill is expected to be signed into law this coming week.
A growing consumer backlash against disposable plastics is driving a change in how water is bottled.
Department stores, zoos, and public facilities in the U.S. and Europe have stopped selling plastic water bottles, and a proposed bill in New York would ban the sale of them at parks, golf courses, and beaches.
Beverage companies are responding by attempting to develop a bottle made completely from recycled plastic, which has been a problem in the past due to discoloration and structural issues. Danone, the company that produces Evian, is betting on a new technology that can hopefully turn old plastics into new containers.
While Evian currently makes only 30% of its bottles from recycled plastic, it has pledged to make all of them from the materials by 2025.
One industry watcher told the Wall Street Journal that the importance of the matter has sunk in because of the broadened awareness of the environment, especially with millennials.
There's been an invasion of the U.S. by a new species of tick from Asia, and it has officials at the Centers for Disease Control worried about the spread of viruses and other pathogens.
The Asian longhorn tick is the first new variety of the arachnid to be found in the country in 80 years. As reported in Entomology Today, it's quickly moving across the eastern U.S. from Connecticut to Arkansas.
The critter thrives in areas of moderate temperature, humidity, and precipitation, which could include a large swath of the eastern U.S. as well as the coastal Pacific Northwest.
The newly detected species is very adaptable and can transmit disease, although to date it's not affected humans. The ticks can attack a cow or other livestock by the thousands and weaken them by depleting their blood supply. The female ticks reproduce without being fertilized, in effect producing clones.
Infections to humans from ticks in general are a big problem. The CDC reported in May that nearly 60,000 tick-borne infections were reported last year, but due to underreporting, they estimate the total number of actual cases may be as high as 300,000.
And finally, it's not often that you hear Dr. Seuss being quoted in federal court, but that's what happened last week when judges reprimanded the U.S. Forest Service for permitting a natural gas pipeline across 2 national forests and the Appalachian Trail.
Repeating the words of the small, fluffy Lorax who tried to save his beloved Trofula trees from a greedy industrialist, a 3-judge appellate panel slammed the agency, saying, "We trust the United States Forest Service to speak for the trees. For the trees have no tongues."
The Washington Post reports that the judges concluded that the Forest Service had raised proper environmental questions early in its permit process, but those issues were suddenly and mysteriously assuaged in time to meet a private company's deadlines.
The court said that the agency's decisions violated both the National Forest Management Act and the National Environmental Policy Act, and found the Forest Service lacked the authority to grant the pipeline a right-of-way across the Appalachian Trail.
The underground natural gas pipeline is being built by Virginia-based Dominion Energy and would stretch over 600 miles across West Virginia, and North Carolina. A spokesperson for Dominion and its partners said they will immediately appeal the decision, saying it will severely harm consumers and do great damage to our economy and energy security.
Lawyers for the Southern Environmental Law Center who brought the case hailed it as a major victory. And if the Lorax could rise up from the pages, he might applaud it as well. Even though he wasn't able to save his fictional Trofula trees, he helped the judges save a real forest